This is component 1 in class question book. Question 1 on photosynthesis. We've got a light dependent stage of photosynthesis, and we've got the Z scheme. That's why it's not called the N scheme, I don't know, but there's the Z scheme. We've got describe the process at A, which replaces electrons lost eventually, and explain how the electrons are used at B and C. So if we were separating this out, we would talk about A, and then we'll talk about B, and then C. So there's 3 marks there. So at A, we're producing these electrons, but where were they? Well, they were in a hydrogen that gets split up into H plus and E minus. Where did that hydrogen? It wasn't supplied as hydrogen gas. It was supplied as water, and then so we get oxygen coming off there. That's not balanced. You'd have to stick a few 2s and 4s in, but that's answering the question: where does it come from? So, water is split to release electrons, protons, and oxygen. And so now we need to move on to get the second mark, how these electrons are used at B. So in B, you can see the electrons are starting at that electron acceptor and ending down in this photosystem. So they're moving from a higher energy level to a lower energy level, and what is that energy being used for? It's being used for ATP synthesis. So the full answer will be it goes down the electron transport chain in chemiosmosis. We've got it fuels the proton pumps, which then protons run through their gradient through ATP synthetase to make ATP. So we could put here fuel proton pumps. And then electrons are used at C. So now these are disappearing off, and so these are used to add to make this hydrogen carrier NADPH. So we get an electron. So these electrons, and where does this H come from? Well, it's these H pluses that we started with. So we're doing this in reverse to make H again. And that is going to be our NADPH. And so of course we need the NADP there. So these components are put together to make reduced NADP or NADPH. Again, you can balance that if you like, but that's answering the question how they are used. They're used to make NADPH. Draw a clear line labelled X to indicate the movement of electrons in cyclic photophosphorylation. So cyclic, photosystem 1, round like that. And so these electrons here, and labelled X, will move round like that. So again, just straight theory there. Part B. The modern representation of the photosystem is shown in the diagram below. Apart from chlorophyll A, name 3 other pigments. Oh, okay, that's okay. So we've got chlorophyll A. So, well, how about chlorophyll B? Chlorophyll B. You could say C there. Carotene, xanthophyll. It's just naming them, and you need 3 to get the 2 marks. State precisely where a photosystem would be found in a plant cell. So chloroplast isn't quite good enough. You need to say where this membrane is. So what's the name of this membrane? It's the thylakoid membrane. Thylakoid membrane in the chloroplast. Then we've got some data, an experiment. We're shining light on these discs, and so light can get in this, and then for some reason they are starting to rise to the top during the experiment. And we've got some darker green ones and some lighter green ones. Calculate the mean time taken for the 15 dark green discs and insert it here. So you're just adding these up and dividing them by 15. One thing to note is, how should you give your answer? So how should you give your answer? Well, look at this other answer and give it in the same style. So we're going to give this to 1 decimal place. And when you add that up, 392.5. So add them all up and divide them by 15. Explain what causes a disc to rise in the solution, and then explain why the dark ones rise faster than the lighter ones. These are connected questions. It's very tempting to put the answer to the part 2 into part 3 and lose some of those marks. So why do they rise? Well, in these discs, they're green. And so photosynthesis is happening, and then, as you know, in photosynthesis we are producing this oxygen. So we get little bubbles of oxygen, possibly, or oxygen filling these spaces inside, and that will make it less dense, and it will start to rise. So oxygen is produced in photosynthesis. O2 is produced in photosynthesis. You can say photolysis there. And O2 fills the gas spaces. In the leaf. It says here, the air in the disc is replaced by hydrogen carbonate solution, and they will sink. So this fills these gas spaces, and you can say, well, it might form bubbles as well. So the disc is now less dense, so floats. So a bit of physics there. Then there's 4 marks. Why the darker green discs rose faster than the lighter green discs. There's 4 marks there. So you need to somehow go from oxygen being produced as a start, and you need to end up with this. So that's faster than the other one. You need to break that down into kind of 4 or 5 steps. So if oxygen makes them rise, presumably one of these gives more oxygen. So at the end of the story is going to be the dark green leaves produce more oxygen. So we can actually put that in right down the bottom. So the dark green leaves produce more oxygen. We don't care now about explaining why it rises. We've said that before. You're not going to get any more marks for saying the same thing again. So you can almost do this backwards. More oxygen produced, so we must have had more photolysis. We must have had more photolysis of water, that's produced more oxygen. Why would we get more photolysis? Well, we must have had more light dependent reaction. So more light dependent reaction. That would be more photolysis, that would make more oxygen. And so now we're actually getting to why we're getting more light dependent reaction. What's the difference between the darker green and the lighter green? Well, the darker green must have more chloroplasts. Well, that's where the green stuff is, the chlorophyll. So more chloroplasts, more chlorophyll. So darker leaves have more chloroplasts, so more chlorophyll. What's the point in that? Well, more light gets trapped and converted into kind of a usable form of energy. So more light is absorbed, more light is trapped and absorbed. And that converts into our kind of high energy electrons that we mentioned before, and transferred to high energy electrons. And that fills our story out. So you can do that forwards or backwards. So darker leaves have more chloroplasts, more light is trapped, so we get more light dependent reaction, so more splitting of water, more photolysis of water, and so more oxygen produced, and that's why they will rise faster. How you would use the technique to investigate the effect of light wavelength. So these four markers, there's been quite a few now in the new style of paper, and they're all pretty much the same structure. You need to tell us what you'd actually change, and that's going to be your independent variable. Quite a lot of students will spend 4 marks trying to get this when it's really only worth 1 mark, and then tell us what you're going to measure, the dependent variable, and then give us 2 controlled variables, and that's how they structure this question. So if you don't know that and try and get 4 marks from describing the independent variable, then you're only ever going to get 1 mark. So our independent variable is light wavelength. Well, how do we do that? So we can change the light wavelength. How are we going to do that? Well, you can do that using special bulbs, or we can do it in the lab using coloured filters, which is the easiest way. Using coloured filters. Or you could use different coloured bulbs. What's our dependent variable? Well, we're going to do the same. Record the time taken for the disk to rise. So record the time taken for the disk to rise. You might think, well, I did that last time, but this is how they're structuring them, so you need to know this. And first controlled variable, well, what did we change last time? We changed the leaf colour, so I'm going to have that as my first control variable. I'm going to keep the leaf colour the same, so keep leaf colour the same, or use the same leaf. And then something else, what else would affect this? If I'm changing the wavelength of light, well, the amount of light I want to keep the same, so keep the light intensity the same. So keep the light intensity the same. So that's going to get us our 4 marks. Other things we could have said, we could of course have said keep the temperature the same. Our source of CO2 mentioned in the experiment is hydrogen carbonate, so hydrogen carbonate concentration, we'll keep that the same. Size of the leaf discs, we'll keep that the same. Species of plant, and so on. So lots of control variables to go at. Predict the results you'd expect from this experiment. Well, this is really testing whether you remember the action spectrum. So you've got the wavelength increasing there, and then you've got the rate of photosynthesis at the side, and your graph kind of looks a bit like that, with red, green and blue colours there. So can you remember that, and just put that into words? So we would expect our red and our kind of blue and red to rise. Would that be faster or slower? Faster or in less time? So less time and more time. So less time would be our faster rates, so red and blue, and so blue, that wavelength, that'll be that way, won't it, for red and blue? No, I'll swap those around. Anyway, doesn't matter. More time for the green, and you can say the colours. I don't expect you to remember all of these wavelengths. So that's 500 nanometers. Red is 650 nanometers. Blue 350. So actually those would be, there we are, right way around now on a proper graph. And so predicting the results. So less time for the disk to rise, for disks to rise. So we're actually relating that to our actual dependent variable, and we've mentioned our 2 versions of our independent variable there.